Hello and welcome to BioRules Video Lessons. Today we are going to be talking about meiosis. But if you talk about meiosis, actually the cell cycle is going to be practically the same if you have mitosis or meiosis. The interface is exactly the same. So you have a G1 phase where the cell grows after having been divided, you have a synthesis phase where you have replication, and you have G2, a growth before the cell divides. And after that, you are going to be having mitosis or meiosis. Mitosis, when the cell wants to make two identical cells, for example, for growing, for replacing tissues or something like that. And meiosis, when it's time to make gametes. The idea of meiosis is to make cells with half the number of chromosomes. So during mitosis, a diploid cell becomes diploid, a haploid cell becomes haploid, a triploid becomes triploid, tetraploid becomes tetraploid. During meiosis, the number of chromosomes is divided by 2. So if you have a diploid cell, that cell becomes haploid. So that's the idea that we're going to be having that. And if the idea is to separate chromosomes, pure simple as that, theoretically we wouldn't need the synthesis or replication phase, because you don't need to copy DNA just to be able to separate the chromosomes that are already there. But because interface always go through G1, synthesis, G2, and then mitosis or meiosis, so even for meiosis there is a replication. And this will have some implications for this process. Okay, so interface G1, interface synthesis, interface G2, it's exactly the same. The cell grows and the chromosomes are replicated. Where you had one chromatid per chromosome, now you have 2 chromatids per chromosome, exactly same, nothing changing here. So, what's going to change? Let's take a look on the steps of meiosis. First step is also called prophase, but this case is called prophase 1. You will realize later that there is a prophase 1 and a prophase 2. So this is prophase 1. So you have a cell that chromosomes have been replicated. You have chromosomes with 2 chromatids each. So everything is ready. And what's going to happen during prophase? Exactly what happens during mitosis. Chromosomes supercoil, exactly the same. Nuclear envelope disappears, exactly the same. The centrioles, when there are centrioles, they migrate to the poles, exactly the same. And the spindles are formed connecting to centromeres. So this here is exactly the same. There is a tiny little difference that you're not seeing here, that you have a consequence in the next one, but you cannot see it in here. So spindles have been attached to centromeres. That's it. So prophase 1, exactly the same. So now let's take a look on the second stage of meiosis. So you have there prophase, chromosomes are already supercoiled, the nuclear envelope disappeared, and things are ready to align chromosomes around the equator. That's what metaphase does, right? So metaphase 1 will align chromosomes around the equator. However, differently from mitosis, where chromosomes are aligned one on top of the other around the equator, the chromosomes during meiosis, they are aligned in pairs. So basically they pair up, chromosome number 1 with chromosome number 1, number 2 with number 2, number 3 with number 3, X with Y or X with X. So they align in pairs side by side. So actually the centromeres here, they have been connected in a slightly different way during prophase. And during metaphase, this difference is visible. So the homologous chromosomes, they pair up and they get aligned in the cell equator in pairs. This is the key difference to mitosis in metaphase 1. This is the one that's different. If you remember mitosis, and you remember this difference in meiosis, you know meiosis, because the rest is going to be the same or a consequence of this difference. For example, if chromosomes get aligned in pairs around the equator, what's going to be happening in anaphase? So anaphase 1 is going to be separating whatever have been aligned. So if the homologous chromosomes have been aligned, what anaphase is going to do is to separate homologous chromosome. So one homologous chromosome to each side, one of the pair to each side, they are separated and they travel to opposite poles. So there they are, you have anaphase. And then during telophase is going to be exactly the same. You have the chromosomes uncoiling, the nuclear envelope being rebuilt, and the cell will go through cytokinesis. So it's exactly the same. See, uncoil over there, and chromosomes, they suffer uncoiling, and you have cytokinesis getting ready to separate. So this here is meiosis 1. And now you could say, okay, look, I have already separated the chromosomes. This cell had 4 chromosomes in the beginning, and now each of these daughter cells have 2 chromosomes. So the job is done. However, because interphase generated the replication and generated the sister chromatids, now we have a problem. The meiosis theoretically is done, but the chromosomes are still doubled, and now you have to separate the sister chromatids, just like mitosis. So because interphase actually isn't planned for meiosis, because you have DNA replication now, after separating homologous chromosomes, you need to separate the sister chromatids. So that's what's going to be happening now. You have the cells that have just been divided, and you're going to have prophase 2. What happens during prophase 2? Now it's all exactly like mitosis. In prophase 2, chromosomes supercoil, nuclear envelope disappears, spindles are formed. If there are centrioles are doubled and migrate to the poles, and the spindles are formed over there. So this is prophase 2, exactly like mitosis. So meiosis 2 is kind of a mitosis. Okay, so all that happened in prophase 2. And now what's going to happen in metaphase 2? Exactly what happens in mitosis. The chromosomes get aligned one top of the other in the equator of the cell. So this is exactly like mitosis, just like mitosis. Okay, if you have the chromosomes aligned one on top of the other around the equator, what is anaphase going to be doing? Anaphase separates. So in this case, if the chromosome is aligned, it's going to be separating the 2 halves of this chromosome. So basically, it's going to separate the sister chromatids. It's going to break the chromosomes into parts. So the sister chromatids, they are going to migrate to the poles. So exactly like mitosis. And what will happen in cytokinesis? You have there cytokinesis. You have the cells that this sister chromatids have migrated to the poles. So now we are going to be having the formation of a new nucleus. A nuclear envelope is rebuilt. There it is, new nuclear envelope for each of those cells. You're going to have the chromosomes uncoiling. So they are kind of ready. And you're going to have cytokinesis again, and the cell gets divided. This here was telophase 2, breaking now the cell. So meiosis was done. Okay. So what will happen by the end of meiosis? So we have there, this is the beginning of meiosis. Actually, you have one cell, 4 chromosomes, each one with a sister chromatid. It goes through interphase, it replicates, all that stuff, and in the end, we are going to be having 4 daughter cells, each of the cell with half the number of chromosomes that we had before. So in this case, you have a green and a pink, Here you have a blue and a yellow, and a blue and a yellow. So if this cell here was diploid, this cell now is going to be, each one of them is going to be haploid. It's 2 divisions, that's why you make 4 cells. This here is meiosis. If we put things here, in perspective, we are going to be having 4 cells each cell with half the pairs of the original chromosome. So you don't have pairs anymore. Now you have 1 of each type of chromosome. Putting everything together, what you're going to be having, G1, you have there prophase 1, metaphase 1 aligned in pairs around the equator, anaphase 1 separating the chromosomes, telophase and cytokinesis separating the 2 cells, and you have now the cells are haploid. The cells have only 1 set of chromosomes, but things carry on. So we're going to have now prophase 2, where you have the nuclear envelope disappearing again, the spindle's been formed, chromosome supercoiling, and now aligning the chromosomes around the equator, and you have the sister chromatids being separated, and you're going to be having telophase, rebuilding nuclear envelope, disappearing the spindles, uncoiling the chromosomes, and having cytokinesis. So in this process, we end up having 4 cells that are exactly half of the original cell. So you have a cell here with 4 chromosomes, now you have 4 cells with 2 chromosomes in each. This is meiosis. Okay? There are some images here that you can take a look with a little bit of calm if you want. We are going to be using this in class to show from interphase until the end of meiosis 2. Here, a diagram comparing mitosis and meiosis. So mitosis and meiosis comparison, both of them go through prophase practically the same way, but in metaphase, the homologous chromosomes pair up around the equator in meiosis, and they don't pair up around the equator, they just go one top of the other in metaphase in mitosis. In anaphase, they separate the sister chromatids, and you're going to be having 2 cells during telophase, and so on. And here, you separate the homologous chromosomes, and then they have to go to meiosis 2, sorry, which is exactly like mitosis. This here is the process of meiosis. Okay? I hope you got the idea. We are going to have a very interesting discussion of this in class, because this is the basis to understanding genetics. Genetics, or what's the chance of getting this or that characteristics, has all to do with meiosis, because it has all to do with what's the chance of receiving this or that chromosome during the meiosis separation. Okay, so now you have time to do the quiz, and I'll see you in class.